I see. Well, in that case, I leave the meeting now. No. So hi everyone, welcome to today's CGDG seminar. Today we have Elena who's giving the talk. Elena is from Spain and she did her undergraduate and, sorry Elena, if I say it wrong, on the Polytechnic University of Valencia in biotechnology. Then she did her master here in the UK in bioinformatics, and at the moment she is about to finish her PhD here at the Rosten Institute in the Division of Genetics and Genomics. And her supervisors are James Prendergast and Albert Hennessa. And yeah, today Elena is talking about her PhD project, and go ahead if you feel ready.
Okay, hello everybody. If you're from the Roslin, you actually might have heard this talk quite recently. I think this is the third time this month I'm talking about this, but I thought I'd take today and potentially talk about a few things in more detail. In the past, I think the audience has been more mixed, and so I've tried to cover things from a more general perspective, but since this is more of a computational genetics group, then I'll probably go in a bit more detail and also talk about some challenges and how we came across them and how we try to solve them as best as we could. So yeah, like Julianne has mentioned, this is the work of my PhD, which is now, I guess, 3.5 years in. And we've been looking into sexual differences in genetic architecture in UK Biobank. Oops, it's not letting me. There we go. Okay, so as you might know, males and females are different. And by different, we truly don't mean just our reproductive function or the way that we look. We mean in regards to a lot of different phenotypes. For example, response to medication or diseases like mental illness or cardiovascular disease. And what underlies these sexual differences is quite a varied a mixture of things, and it's truly not simple and not very well understood at all. You might be thinking, oh, well, you know, there's the sex chromosomes. Males and females have a different set of sexual chromosomes. And that's true because the males have a particular set of genes within the Y chromosome that females do not have. While genetics is very important in sexual differentiation, once a human is already fully developed, there's a lot of different factors that could also be contributing to the differences that we're observing. For example, we know that males and females don't have the same DNA modifications. So epigenetics differences could also participate in differences in the phenotypes that we're observing. There's also differences in genome regulation between the sexes, as well as differences in hormones or potentially even sex-specific regulatory networks. And then something that people might not even think of is that there are differences in exogenous factors as well. So because males and females have different gender roles in our society, it could be that, for example, males are more predisposed to a particular environment or particular working hazards, while females are more predisposed to other environmental exposures. And these could as well lead to these differences that we're observing. Now, here I've highlighted genetics because that is the focus of this study, and specifically genetic architecture, which you might probably already know, but it's just a fancy term to say anything that is related to your genetics that could be leading to the phenotype. So this could be anything from relationships between different genes, between different variants, to a gene-by-environment interaction. And in fact, talking about gene-environment interactions, what we are discussing here is something called a gene-by-sex interaction, whereas the environment is defined by our sex. So you probably know this equation, the genetics plus environment equals phenotype framework, by which your genetics is one thing and then your environment is everything else. And here we're talking about your environment as defined by factors that are related to your sex. So things like differences in epigenetics, things like differences in genome regulation and hormones, so on and so forth. All of these things are englobing our genetics and could potentially be interacting with our DNA to lead to some of these differences in phenotype. And this is what we are calling a gene-by-sex interaction, which is the focus of this study. So there are different ways of studying gene-by-sex interactions, and they all make use of classic quantitative genetics methods, which I've arbitrarily divided into the broad view and the narrow view methods, just based on whether we are looking at a particular portion of the genome or whether we are looking at the genome as a whole. So in the broad view methods, I talk about the methods that we use to kind of assess whether there is a difference in gene by sex, whether there's a presence of gene by sex interactions in a phenotype, by quantifying the relationship of the genome as a whole to the phenotype. So for example, we could obtain heritability calculations or heritability estimates, which measure how much of the phenotypic variation is due to genetic variation. So if we were to calculate heritability in males and females separately, this would then allow us to compare these heritability estimates. And if a difference was found, we could assume that there is a level of divergence between the sexes in regards to genetic architecture. There's also genetic correlations between the sexes that could provide insight into the presence of gene by sex interactions. So like the name indicates, a genetic correlation is essentially a measure of how correlated the genetic control of a given phenotype is between either two traits or two groups. Here we would be considering the same phenotype on different sexes. And so a large genetic correlation would indicate a large level of genetic control. Okay, and then moving on to the narrow view methods, I think this is potentially what's more popular nowadays. It's the genome-wide association studies. And what genome-wide association studies allow us to do is to dissect the genome and analyze each individual genetic variant and identify its association to the phenotype. So by performing a sex-stratified genome-wide association study, we are able to estimate the association or the effect of each individual genetic variant to the phenotype for females and for males separately, and then compare whether this association differs significantly or not. So if we look at the literature, we will find that differences have been found and gene by sex interactions have been found, but they're not very widespread. They're not very big. I think the overall consensus was that we needed more statistical power and to study a wider breadth of traits, which was not done until UK Biobank allowed us to start performing these very big studies as well as a very big breadth of traits, as I'll mention in a second. And we also need to understand the phenomenon because we truly don't really understand what underlies these gene by sex interactions, like is many times the case with a genome-wide association study. So the aims with the study were, first of all, to establish whether gene by sex interactions exist, locate them across the genome, but then also not just stick to that because, like I mentioned, genome-wide association studies many times act as a black box by which you find an association to a phenotype, but you don't really know why that association is showing up in your analysis. I said that it's being mediated through an effect in the transcriptome. Is that is there an underlying biological pathway that's acting? We don't really know, and so we wanted to potentially shed light on that as well. So our data here is UK Biobank. If you don't work as a human geneticist, you might not know it, but I guess it is quite well known. UK Biobank is a population cohort consisting of around 500,000 individuals, which are aged approximately from 40 to 70 years old at recruitment, I believe. And it's a pretty healthy cohort. And this cohort was both genotyped and extensively phenotyped. So this allows geneticists to obtain a wide amount of genotype-phenotype information. And so in this particular study, we are focusing on around 450,000 individuals from UK Biobank, as well as 530 different phenotypes, of which we have both non-binary and binary phenotypes. And by non-binary and binary, we just mean non-binary continuous traits like height, for example. And by binary, we mostly mean disease traits, so case control traits, whether you have had the disease or you've not had the disease. And then after quality control, we have around 9 million genetic variants left. So I'm not going to go too deep into the methodology, but essentially what we did and what underlies a lot of this work is sex-stratified models. And what we did with these models, or the objective of running models, is essentially to quantify, again, like I mentioned prior, quantify the relationship of both your genome to the phenotype, as well as quantify the relationship of each genetic variant to the phenotype. And so we did this separately for males and for females, and thus we were able to obtain or dissect the phenotypic variants for both males and for females. And we did this by running linear mixed models, which accounted for population structure. And so what we were able to obtain from our linear mixed models was essentially both, again, variance components to the phenotypic variance. So we were able to obtain estimates of the additive genetic variance for each of the sexes, as well as the environmental variance for each of the sexes. And now this quantifies the relationship, going back to the broad view methods, this quantifies the relationship of the genome to the phenotype as a whole. And then we were also able to run a sex-stratified genome-wide association study, which allowed us to obtain genetic effects for each of the genetic variants across the genome and obtain a sex-stratified genetic effect estimate, so a genetic effect for females and for males separately. And of course, we did this for the 530 different phenotypes that we are considering here. So before moving into the results, I wanted to address this. And so you might be thinking, why are you guys running a linear mixed model instead of a logistic model for a lot of these traits when they are binary? And classically, logistic models have been the preferred tool of choice for binary traits. And we still ended up running linear mixed models. So why did we do that? Well, first of all, the software that we were using ran linear mixed models, and it was just easier for us, as well as logistic mixed models have become more popular in recent years. But again, going back to the linear mixed models, the problems that linear mixed models have with binary traits in these kind of studies is that they can lead to false positives when the case control imbalance is large. And this is the case in UK Biobank, because again, it is a pretty healthy population cohort. So cases were not very, they were not very large in a lot of the different phenotypes that we were considering. So if you look at this paper that I've included here, they ran some simulations and they found that if you do use a stricter math threshold to ensure that the minimum allele is well represented, then the problem does seem to be offset, and the false positives do not appear anymore. So that is essentially what we did. We used a math 10% filter for the binary traits to account for possible false positives. And so we ended up considering, in the genome-wide association study, we ended up looking into 9 million variants for the non-binary traits and around 4 million variants for the binary traits. And now going back to the logistic mixed models, when we turned our paper into the reviewers, they did suggest us running logistic mixed models to make sure that we were still not capturing any false positives, as well as they wanted us to argue why we should be using linear mixed models as opposed to logistic mixed models. And I won't go into the theory basically because I'll butcher it. But if you are interested, here I've included a paper that does talk about it. It's called Logistic Regression, why we cannot do what we think we can do and what we can do about it. But essentially, logistic mixed models are great for case control phenotypes. But in this particular case, where we want to compare the genetic effects of two different samples, males and females, logistic mixed models are not really appropriate because you first would have to transform the genetic effects, which are not at the same scale in each of the different models, prior to comparison. And there are different methods to do this, each with their own different caveats. Either way, we ran our logistic mixed models, and to do that, we used Regini. I tried to use Sage, which some of you might know, but it was a bit slow for our use. So it was suggested to me that we use Regini, and it didn't work very well. So we used Regini, and we found that our results replicated very well. So that's just kind of like a problem that we ran across that I wanted to kind of mention if you are in a similar situation. Okay, so moving on to the actual results, did we actually find gene by sex interactions? Sure, the short answer is that we did. So first, we looked into differences in heritability, which again is the fraction of the variation of the trait that can be explained by the genotype. And we found that 30 of the 446 binary traits that we looked at had a significant difference in heritability. The non-binary presented around 50% of them presented a significant difference in heritability, which is quite a large proportion. We also looked at whether these differences in heritability were being driven by differences in genetic variance as opposed to differences in environmental variance, because we know that heritability includes both of them in the denominator. And so here I've plotted the log two-fold change of male to female variance, just to see whether the genetic variance difference was the one that was largest, and it wasn't the environmental variance that was actually driving the difference. And so here I've included the 5 traits that had the smallest comparison p-values, and you can see that yes, that is the case. And here in these kind of donut plots, I include the proportion of the traits, or the binary and the non-binary traits, that also had a larger fold change in genetic variance as opposed to environmental variance. So you do see that this is the case for the majority of traits that had a significant difference in heritability between the sexes. We also looked at genetic correlations that significantly differed from one. Again, this being in lay terms, the common genetic control between two groups. And for this, we used LD score regression, which uses GWAS summary statistics to obtain the genetic correlation estimate. And a bit of a con here is that it requires that your binary traits have at least 5,000 cases, so that they narrow it down for us from 400 and some traits to 77. But even still, we found that 11 of these 77 binary traits did have a genetic correlation that significantly differed from one. While for the non-binary traits, it was again a larger proportion, with 58 of the 83 having a genetic correlation that significantly differed from one. Here I've included a histogram, which shows for these traits that had a significantly different from one genetic correlation, trying saying that 5 times. You do see that the genetic correlations do cluster around the 1 mark, meaning that genetic correlations are quite close to 1, even if they do significantly differ from it, meaning that genetic control is very similar between the sexes and these phenotypes, and that kind of mirrors what's been stated in the literature. However, there are a couple traits that do differ and they're further away from the 1 line. So things like disorders of urinary tract, hernia, eczema, gastric reflux, waist-hip ratio, so on and so forth. We also compared our genetic correlation estimates across studies, just as a sanity check. So we were more limited in regards to the traits because it is that more anthropometric traits have been the study of sexual differences in genetic architecture as opposed to other phenotypes. So here I've plotted for 6 different phenotypes, the estimates of genetic correlation across study. Our own study is the one here labeled in pink. And then we have 3 other different studies. And you do see that our estimates all lie within the confidence interval of the other studies, which is good to know. And then moving on from the broad view methods and moving on to the narrow view methods, or the sex-stratified genome-wide association study, which I think is potentially the most interesting part. We did find a very large proportion of traits that had at least 1 variant that had a significantly different genetic effect estimate. So 61 of the 84 non-binary traits presented at least 1 sexually different variant. We see that waist-hip ratio is the clear winner here, with 100 different independent SD SNPs, as we are calling them. And the binary traits, we were again limited in twofold. We were limited in regards to statistical power because we do depend on the number of cases for these diseases. And we were also more limited in regards to the number of variants because we did have a MAF 10% cutoff. But even still, we did find interesting things. Here we have ankylosing spondylitis with the largest number of independent SD SNPs, 18. And then we see things like gout or hypothyroidism popping up as well. Here I've included the Manhattan plot for waist-hip ratio because it is the star trait which has been described in the literature the most often. And so if you don't know what a Manhattan plot is, it's essentially what you see right here, where the x-axis is the genomic position. I've marked the different chromosomes, and they are also colored differently just to see the boundaries better. Each point is a SNP, and the height of each of the SNPs represents the -log10 p-value for the statistic of the sex difference. So here I've included the statistic that we used as well. It was essentially a t-test that compared the genetic effect in males and in females. And so the smaller the p-value, the higher the SNP will be here, and we get these peaks. And you can see that there's not really an area in the genome where you are getting more of these gene by sex interactions. It is pretty well distributed. And this was the case for a lot of the non-binary traits, where you do see that they are, I don't want to say polygenic, but they are polygenic. But what I mean in regards to gene by sex interactions, they are very well distributed across the genome. And this was also the case. It wasn't just the case for non-binary. There were a couple of diseases that also showed this behavior, while other diseases did have fewer peaks. So those that seem to be more related to immune response that have a very big peak in the HLA region, as opposed to other areas of the genome and so on. But it was very much a trait by trait case. And yeah, here I've also included on the right a little comparison. So when we ran the study back in 2017, 2018, when I first started my PhD, we were the people with the largest sample size. But since then, others have published an even bigger study where they did a meta-analysis of both UK Biobank and Giant, and they had around a sample size of 600,000 or 700,000. And so they obtained a very large number. They basically obtained very similar peaks to us. And here I've actually directly compared our sex comparison p-value from this test statistic. And I ran their genetic effect estimates through the same equation. And you can see a very, very good correlation between the two, which at least serves as a sanity check for us. Okay, so up until now, I've been talking about the existence of gene by sex and how we found evidence of gene by sex across the genome and across phenotypes. But what can we actually do with this? What are the consequences of gene by sex? So is it possible that these differences are leading us to miss things when we are considering males and females together within a genome-wide association study? So for example, it could be that a genetic variant is interesting in just one sex and not the other. And by considering them both together, this added extra noise leads to a non-significant signal in a non-stratified effort. Or for example, in a more extreme scenario, it could be that a genetic variant has a negative effect in one sex, positive effect in the other. And thus by combining the two in a single model, you obtain a resulting genetic effect close to zero, and you end up not detecting it. So this is what we wanted to explore. And so the first thing that we wanted to see was if there is a significant difference, could we potentially significantly improve prediction of phenotypes by using sex-specific genetic effects? And also taking it even further, are we potentially missing SNPs of interest by combining both males and females together within a single model? So kind of like what I've just described here with the negative and the positive, or the significant, non-significant. So first talking about the improvement in prediction, what we did to test whether we could obtain an improvement in prediction by using sex-stratified genome-wide association studies, what we did is we took a subset of UK Biobank, which is the unrelated white British individuals, and we divided them into two, the training and the testing data set. The training consists of around 300,000 individuals. And what we wanted to do with this training was essentially recalculate genetic effects for a male-specific model, a female-specific model, and a whole population mixed model. And what we wanted to do here was to match sample sizes. So that's the only reason why we reran our models, as opposed to just using what we already had. And so we ended up running a GWAS with 150,000 females, 150,000 males, and the mixed effort had 75,000 of each. And now, having obtained the new genetic effect estimates, we proceeded to obtain our predictions. And we did this in the form of polygenic scores. And polygenic scores are quite simple to calculate. If you don't know what it is, it's essentially just taking, for the SNPs that you want to use within the prediction, you take their dosage information and you just multiply it by the genetic effect that you've estimated in your genome-wide association study. And then you just sum all of that up. And so to obtain our polygenic scores, we did this in 3 different ways in a way that allows us to then compare whether we obtained a better prediction or not. First thing that we did was obtain the same polygenic score, by which we obtained the estimate of the polygenic score using the genetic effect for your corresponding sex. So if you're a female, then we would obtain your prediction using genetic effects for females. Same if you're a male. And then we also did this the other way around, so obtaining an opposite polygenic score. If you're a female, then you would obtain your polygenic score with the genetic effects that we've obtained in the male-specific model and vice versa. And then finally, we have the agnostic polygenic score, by which, independent of your sex, we would be using a genetic effect that's been estimated in a sex-agnostic model. So this is what's currently being done when you obtain a polygenic score, or for example, where 23andMe returns a prediction. What they're essentially doing is obtaining polygenic scores, and they're using genetic effect estimates from a sex-agnostic model. So what we found is that we found no statistical difference between the same and the agnostic polygenic scores. However, we did see an increased trend in prediction accuracy, which we measured as the correlation between the polygenic score and the actual phenotype value in the testing data set. And here I've plotted this here. So here you have the correlation between the opposite polygenic score and the phenotype, and then the agnostic and the same for males and for females. And you can see this trend going upwards in all of the tested traits. So just the non-binary, the binary that we did test with at least 10 SD SNPs basically showed no difference, but it was also more difficult to measure. So yeah, we did think this was quite an interesting finding, and it could be a point to make in the future to use sex-specific models to predict phenotypes. So again, going back to the slide a couple slides ago, where I was talking about both the prediction as well as the masking, we next moved on to perform our masking analysis, by which we wanted to see if there were SNPs across the genome that were significant for at least one of the sexes, but were non-significant in a sex-agnostic model. So for example, you could find a genetic variant that is significant for one sex, for example, females, non-significant in the other. By considering them both together, you end up with a non-significant hit. So we did this across the genome and across phenotypes, and we found a very large proportion of traits that had at least one potentially masked SNP. So it was around 98 of the 446 binary traits that presented at least one of these SNPs, while it was even larger for the non-binary, with 78 of the 84 presenting at least one masked SNP. So again, this could be a good argument for people to, in the future, run not only sex-agnostic models, but also sex-stratified models to make sure that they're not missing anything, make sure they're not missing potential interesting SNPs for just one of the sexes. Okay, so now we move on to our functional analysis, or how we tried to shed light into what all of this means. Why are we finding these gene by sex interactions? What is actually happening there? What actually happens within this black box by which we are finding a gene by sex interaction? Is there a particular pathway that is enriched in all of these SD SNPs that we are finding? So to do that, like you probably might have guessed, we did a gene set enrichment analysis, by which we looked for gene sets that were potentially enriched in all of these SNPs that we were finding. And so to do that, what we did is we took software Magma, and we took our SNP data and we translated it to gene data. So briefly, what Magma does is it takes all the nearby SNPs to a gene, and then it assigns a combination of the data of these SNPs to the gene. So you end up going from a dimorphic SNP to a dimorphic gene as a combination of the dimorphism p-values that we had obtained in these nearby SNPs. And with that, we were also able to split the genes into those that had a larger effect on average in women and those that had a larger effect in men. This, of course, was done per phenotype. And then with those different sets of genes, we did set enrichment. And we did that with FUMA, which is a web-based software. And so I'm not going to go too deep into it, but I will mention what is potentially the most interesting finding from the gene set enrichment analysis. And it's that we did see, when we obtained our heat maps, we did see a cluster of gene sets relating to miRNA regulation for some of the female dominant genes in body mass traits, which we thought was interesting because in the past, miRNA has been postulated to be a potential regulator in all of these differences due to its characteristics. But again, this is very generic, and more studies need to be done to truly see what this actually means. And then finally, the last thing that I'm going to be talking about today is how we also wanted to potentially see if these gene by sex interactions were arising due to differences in gene expression regulation. So what we mean by that is it possible that these sexually different SNPs are close to a gene, and a difference in genotype in this particular SNP could lead to a difference in gene expression regulation depending on your sex. So for example, could it be that this SD SNP binds to a sex-dependent factor And so a difference in genotype here would indicate that this sex-dependent factor might have a different ease as to its binding to this particular site. And thus, this could regulate gene expression in one of the sexes because it is sex-dependent, and we would obtain something like this. And this is what we are calling a sex-biased eQTL. And so to do this, to analyze whether there were areas in the genome where we could observe this behavior, what we did was we obtained we took all our SD SNPs and we looked at the nearby genes within 1 megabase window. And in order to run our models, we shifted from UK Biobank and we moved on to GTEx. GTEx is another cohort which focuses less so on the phenotyping of individuals and more so on transcriptome data. And it does so across a huge number of tissues, as opposed to many times some of these analyses focus more on blood phenotype or on blood transcripts. And here we are talking about 39 different tissues, which is pretty large. The main inconvenience of GTEx is, I would say, the sample size. We only have 450 individuals with the version that we were using. We have 39 tissues, like I said. However, the number of samples per tissue does vary quite a bit. It goes from 72 to 361. And again, the number of transcripts does vary, but this is just a question of the tissues and how each tissue has a different transcript profile. And so the models that we ran to establish whether we could see these relationships essentially were just a linear model where we included an interaction term with SNP and sex. And this would then would represent this difference in slope that we are observing here. And we did this for each of our pairs, each of our SD SNP gene pairs, as well as each of the 39 tissues. So in the end, we ended up with 8 sex-biased eQTLs across all of the different tissues and across all the pairs that we tested. But overall, larger sample sizes are needed to truly uncover whether there is a link between these gene-by-sex interactions in our GWAS, as well as gene expression regulation. And a recent study did say that in order to truly uncover sex-biased eQTLs that could be arising or that could be then being found in GWAS the way that we found them, you would need far larger sample sizes than what are presently available. But even so, I am going to be talking about one of the sex-biased eQTLs that we found, kind of like an example of how this kind of framework could potentially help us to uncover potential biological pathways that could be underlying some of these gene-by-sex interactions. So again, this is one of the sex-biased eQTLs that we found. We found it in the muscle skeletal tissue. And here, the SNP that we found to be a sex-biased eQTL is this one. I'm not gonna try and say it because I'm pretty sure it won't mean anything to anybody just because it's a random number. But the gene that we are considering is MICA, and that is in chromosome 6. And here you can see the genotype. So the SD SNP genotype, GG, GA, or AA. Each point is an individual colored in by your sex. And the y-axis is gene expression. And you can see a slight difference in slope that we did find to be statistically different. And so what I found particularly interesting about this SD SNP gene pair was the following. I'll just take you through my thought process when I was looking into the results. Essentially, this particular SD SNP we found to be sexually differentiated. We found it to be an SD SNP for ankylosing spondylitis. So it was differently associated based on our sex to this particular disease. And this particular gene, MICA, has also been linked to ankylosing spondylitis as well as to other similar diseases. So that in itself, I thought was quite interesting. And so when I saw this, one of my supervisors, James, he suggested to potentially look at CheapSeq analyses, CheapSeq databases to see if any study had linked any protein to be binding to this particular area of the genome. And I did find in one of these databases that FOXA1 did seem to be binding to this particular SNP. And so I thought that was quite interesting, because if you look into the FOXA literature, you'll find that it does seem to be modulating transcriptional activity of nuclear hormone receptors. therefore, you could say that FOXA1 binding could be influenced by estrogen levels or another hormone level, thus leading to a difference in its binding to this particular SNP, could then lead to a difference between the sexes in gene expression regulation of MICA, and then even further downstream to a difference in susceptibility. But of course, this is just a story time like I've written here. But it is quite interesting to see how larger sample sizes do become available, whether we could potentially start filling in the holes of where these gene by sex interactions come from. Okay, so I think that's it for me now. I'm getting a bit of a sore throat from talking so much, but essentially the conclusions are that we did find sexual differences in genetic architecture for a large amount of traits, though it was of generally modest magnitude. While we found no statistically significant improvement in prediction, we did see a consistent increase in predictive accuracy. We also found potential masking of loci of interest in sex-agnostic models, and we need larger sample sizes to truly uncover links between SD SNPs and sex-biased eQTLs. So as an acknowledgement, of course, my supervisors, both Albert and James, as well as to all my team members, because it does take a village. And yeah, so thank you for listening. If you have any questions, as well, I'll also leave the preprint to the paper. It's been up since last summer. It's currently under revision. But yeah, if you are interested, just have a look at that. It will cover basically most of what I've said. But yeah, if you have any questions, do let me know.
Thank you, Elena, for this really, really interesting talk. I haven't seen any questions yet. So if you have some, either use the chat function, or I think you can just unmute yourself if you would like to speak yourself. In case there are none yet, or you need some more time, I mean, I have plenty of questions. When I just start with one, okay? We talked about the potential masking, and you demonstrated it really nicely in your simulation studies. So what would be the take-home message, or what would be the solution? Would you suggest to perform, for example, GWAS now within the sexes and then compare? Because obviously, I think when you have a GWAS on for both for females and males, and then just fit the sex, you still have this masking problem, right? Or
yeah, so what I would do is, I mean, it makes sense to run males and females together, especially because we do see that the majority of genetic architecture does seem to be shared. And by cutting them in half, you would essentially be cutting your sample size in half, which would then lead to decreased statistical power. So what I would do is I would do everything the same. I would still run males and females together within a model. But then beyond that, I would potentially also perform sex-stratified genome-wide association studies just to make sure that you are not missing anything. So it's kind of best of both worlds, really, where you run your sex-agnostic model with your huge number of samples. And there is evidence to suggest that you won't be missing much. But if you are missing something, just to make sure, just run the model stratified by sex as well.
And is there any, because you obviously talked about humans, is there anything known in literature and livestock species?
I'm not sure I could comment on that. I don't think I have enough information, but I don't see why not. I think, especially mammalian species, we are all much more similar than we think. So I personally, if there are sufficient differences in phenotype between males and females within a species, I think it would be the same thing. You would find that there could be sex-specific factors that could be interacting with our genome. So I would say that, yeah, it's probably worth looking into. I guess it also depends on what you are looking to do with your genome-wide association results.
Yeah. And anything yet? I mean, I can continue if people don't want, but okay. I haven't seen a question yet, so I just continue. I think that's fine. At the start of your talk, you talked about how much variation is contributed to the sex effect, and how much is basically probably environmental. But might there be still a confounding? Because I think you haven't fitted any environmental factors in your model, right? You just fitted the sex.
No. So our models, we're not actually fitting sex. We are running males and females separately. But in our covariates, we are fitting a lot of things. We are fitting principal component, like 20 principal components. We also have a polygenic effect that accounts for population structure. So truly, we are accounting for a bunch of different things. So this should not lead to a difference in an environment between males and females that we are not accounting for, not an obvious one at least.
Yeah, because no, I was just, I missed this obviously.
It's okay.
And then I was just surprised that you have this huge contribution, or because I think if you wouldn't fit any environmental factors, you wouldn't see this huge proportion of variance explained by sex as opposed to other factors.
Well, there is like I mentioned, there are differences in environment between the sexes, which it could be that males and females do have a difference in environment that could also be leading to these differences. So, truly there is still so much more to uncover. We really have just scratched the surface with this.
So which model was actually used to calculate variance components? I mean, obviously here you have separated models by sex, but you also need to include covariances, right?
Yeah. So it was essentially this. We did it in 2 steps, by which, first of all, we obtained the variance components using this model. Just ignore the SNP portion. It was the same covariates, 20 principal components, assessment center, and a bunch of other things. And then you obtain the variance from the polygenic effect. Here you would obtain the variance component for your genetic variance component. And here you would obtain from the E factor or the residual factor, you would obtain the environmental variance component. And then having fit that with the residuals, we then ran the actual genome-wide association study. So the residuals were used as a proxy for the phenotype, having been adjusted for the covariates. So it wasn't all run at once. We did it in a 2-step way. Does that make sense?
Yeah, okay. I'm not sure there's anything wrong with my chat, or I mean, Letizia and Ivan, if you see any questions, just point me. I just have
it seems that people are shy today. I don't know why.
Oh, it was so well explained, and I was the only one who missed some bits anyway. Just one more thing, Elena. When you really nicely explained the problems you faced with the logistic model and the linear model, you mentioned obviously that you would run into problems with the linear model once the case control imbalance is large. Can you somehow quantify large? Are we talking about 5% cases, 95 controls, or is there something like, I mean, I think it's probably not really black and white, but maybe there's something like a recommendation you can give us.
It's totally quantifiable. So here, these guys, they ran simulations where they obtained, and they gave you kind of the type 1 error rate based on both the cohort sample size, as well as the number of cases that you have and the math threshold that you're using. So it's a very nice table if you're interested or you're coming across something similar, where you can kind of see the math threshold that you would need to use to make sure that you're getting rid of that inflated type 1 error rate based on your cases and the number of controls that you have. So for our case, because we're using minimum of 400 cases and we have around 200,000 individuals for each sex, this is the threshold that made sense for us, but it depends on your cases and your controls.
Okay, and I'm sorry, I have one more question. now that I've seen this, it's probably because so you said with the binary traits, you sometimes run into problems if you had the high imbalance or missing data. And sometimes in GWAS you use, you have a quantitative trait or a continuous trait, and you basically use just the extremes to basically convert them to case and control, if this makes sense. I mean, obviously, if you just have
like tall or not tall.
Or very tall and very tiny.
Yes. Yeah.
So basically, and sometimes you can increase the power if you basically use very extremes. And would this basically may have sense for yours as well to, for example, I don't know, let's take like obesity, obese, yes or no, and you have a lot of missing values, would have make sense to use a related trait like body mass index, which is probably quantitative, and somehow group individuals into
at first I'm not
into
exactly
categories, but
I'm
maybe
not
yeah.
sure. I'm just thinking that it's just the continuous nature of our non-binary phenotypes made it so much better for us
Yeah.
because we obtain so much more information.
Yeah.
I'm not exactly sure why putting them into category. I'm pretty sure they wouldn't
No,
really give us.
I was just wondering, in I suppose when you have binary trait with very unbalanced phenotypes, let's say
Yeah.
a lot of non-obese people, hopefully, and a tiny proportion of obese people, to instead of just skipping this trait. I mean, okay, you didn't skip it, but yeah, maybe use BMEs and category. But anyway, I just wondered about that. But just ignore this question. Okay, so if I don't have any more questions, and I don't see anyone else. Oh, there's one question. Sorry.
Can't see the chat.
I can read it if you want. John is asking that, or he is stating that with a minor allele frequency of 0.1, you lose many variants that may be causal. How many SNPs were filtered out? If logistic regression gives similar results as LMM, why not use LR and potentially a lower minor allele frequency threshold?
Okay, that's a very good question. So like I mentioned, we had already run our linear. He basically made the same point that the reviewer made in the paper. So the first question, I think, is how many we filtered out. So that's exactly what I have here. We ended up with around 9 million variants for the non-binary traits and around 4 million for the binary traits. So you do see a pretty big reduction. In regards to why we wouldn't use logistic, like I mentioned before, we would have to transform genetic effects prior to comparison, and each of these methods have different caveats. We ran one of the transformation methods, and we saw that it didn't work very well when maths were lower, and it had a couple, it wasn't perfect. And either way, we saw that the results replicated really well with our linear mixed models, which led us to believe that it really wouldn't be worth the effort. We weren't missing basically anything with the logistic mixed models. We ran these models without the filter, and we weren't coming up with sexual differences in the SNPs that we had discarded. So yeah, I mean, it's an option, but we would have to look further into these transformation methods to make sure that we aren't putting in, I mean, it's not like the logistic mixed model is perfect. It's essentially what I'm trying to say. And we found that the results were very similar to our own linear mixed models, which made us argue that in favor of the linear mixed models, essentially because we already had the results and we didn't see that we were going to gain much from the logistic mixed models. Now, if you are going to run a study from scratch, I mean, you can potentially look into the logistic mixed models, but knowing that you shouldn't be comparing them directly, and you would have to be looking into transformation methods.
Okay, thank you. I think this answered the question very well. Yeah, he says yes, but he has another question. So basically, the prediction accuracies you showed look very low. Is this common in human genetics?
Right. So I should have, I'm not sure if I made a point of that, but we are only using, oops, I am lost within my own slides. Yeah, we are only using the SD SNPs. So we're not doing a prediction like you usually would with all of the genetic effects across the genome. We're only using the prediction using SD SNPs, like I'm stating here. So because SD SNPs are not that, we don't have many. There were some of these traits where we were only using 10 SNPs for the prediction. We actually don't expect a very good prediction. We were mostly interested in the differences, if that makes sense.
Yes. Okay. Anyone else? Nope. Okay, then thanks again, Elena, and for everyone else, and also for you, Elena. We have the next talk in 2 weeks, and we will hear from Matea Yanis on a web-based selection tool for pedigree dogs. Okay, bye.
Bye.